Good afternoon, everyone. Today we are going to present our project titled Assessment of Soil Improvement through Vermicompost using Basil Plants. This study focuses on how vermicompost affects plant growth and nutrient uptake compared to normal soil. Vermicompost is an organic fertilizer produced by the decomposition of organic waste using earthworms. It's rich in essential nutrients such as nitrogen, phosphorus, potassium, calcium, and beneficial microorganisms. Vermicompost improves soil structure, water reflection, and nutrient availability for plants. Excessive use of chemical fertilizers can damage soil quality and harm the environment. Many soils lack of sufficient nutrients needed for healthy plant growth. Therefore, there is a need to find a sustainable and eco-friendly alternative to chemical fertilizers. The main objective of this study is to evaluate the effect of vermicompost on basil plant growth. We compared plant growth, biomass, and nutrient content between control plants and vermicompost-treated plants. Our hypothesis says that vermicompost will improve plant growth and nutrient uptake compared to the control one. The experiment was designed as the following. Total of 6 pots, 3 of them are control without a vermicompost, and the other 3 pots are treatments with vermicompost. We planted 2 plants per pot, 12 plants in total, and the irrigation was 100 ml 3 times per week. The duration of the experiment was 2 months approximately. Regarding the harvest and plant analysis, shoots and roots harvested are separated. Fresh weight has been recorded for all samples before and after. 4 samples have been selected for nutrient analysis, extracts analyzed for potassium, calcium, and phosphorus. The results show that plants treated with vermicompost has longer shoots and roots compared to the control plants. This actually indicates that the vermicompost positively influenced plant growth and root development. A one-way ANOVA test was used to determine whether the difference between control and treatment group were significant. The results actually showed that there is a significant difference in shoot and root length, confirming that the vermicompost improved plant growth. This graph shows the effect of 2 treatments on plant growth. The orange bars show higher values than the blue bars in all parameters. Fresh shoot and fresh root weights are higher in the treated plants. Dry shoot and dry root weights are also higher, showing real biomass increase. Arrow bars as shown represent variation among samples. Overall, the treatment improved both shoot and root growth compared to the control. This table shows the statistical comparison of fresh weight and dry weight between control and the treated plants. Although the treated plants showed a higher biomass, the difference is not statistically significant, possibly due to a small sample size and short experimental duration. Potassium content was higher in both roots and shoots of vermicompost-treated plants. Potassium is important for plant metabolism, enzyme activation, and water regulation, so this increase supports better plant growth. As we can see here, calcium levels were also higher in the treated plants. Calcium actually plays a key role in the cell wall strength and root development, which helps plant grow stronger and healthier. Phosphorus content increased significantly in the treatment group. Phosphorus is essential for energy transfer, root growth, and overall plant development, explaining the improved growth performance. Vermicompost clearly improved basil plant growth and nutrient uptake. The increase in potassium and phosphorus was especially noticeable. These improvements are likely due to better soil structure, increased microbial activity, and slow nutrient release. The lack of significant biomass difference may be due to limited samples and short study duration. In conclusion, vermicompost significantly enhanced shoot and root growth. It also improved nutrients uptake in basil plants. Vermicompost actually shows a strong potential as an effective organic fertilizer. This supports sustainable agriculture and reduces dependence on the chemical fertilizers. At last, future studies should include a larger number of samples. Different vermicompost application rates should be tested, and the experiment duration should be extended. Soil chemical properties should also be analyzed for better understanding.